Hello, a warm welcome to all. Today we are going to talk about another important topic, which is called the sources of acids in human body. So in the last two classes we talked about acids and bases, and readers can go to my classes. For a quick recap, what I thought was that we define acids as substances which donates hydrogen ions, and we measure the strengths of acid with the help of our pH, which is nothing but negative of log of hydrogen ion, and this pH determines the strength of the hydrogen ion concentration, and the normal pH in blood is 7.35 to 7.45, which corresponds to the hydrogen ion concentration of 40 nanoequivalents. This was the last class we did. So what are the sources of these acids in human body? So this is the topic that we are going to discuss today, so let's dive in. So the human body produces acids from two sources. One is number one, from normal cellular metabolism. So what is a cell then? Cell is the smallest and the basic building block responsible for carrying out life processes in human. The smallest and building block. So as you see, we all learn in biology classes, this is a cell. This cell consists of a nucleus and the cytoplasm, and there is a cell membrane. Now there are a lot of cells in the human body. So these cells, brain cells, liver cells, blood cells, and these cells, when they get together, they form tissues and organs. Now through cellular metabolism, the cells perform very important function in the body. For example, they help in formation of enzymes and hormones. For example, insulin is a hormone, so when you take a meal, insulin helps to regulate the blood glucose level. So also when you take food, the pepsin is secreted by the stomach, helps in digestion of the food. Secondly, they help as the movements of nutrients to and from the cells. So glucose is transferred to and from the cells, so also is water. Thirdly, cells undergo a lot of cell division and maturation, new cells are formed. So all this process requires energy, and when this energy is utilized, acids are formed. So let us go how this is really formed. So second way of formation of acid is the most important. It's a breakdown of nutrients in the body. So what are nutrients? And nutrients are substances which the body needs to function and stay healthy. So these are substances which the body needs to stay healthy. So what are the nutrients we commonly take? Number one is the carbohydrates, bread, rice, we already take regularly in our meals. Proteins like egg, fish, meat. Fats like nuts, eggs, and all. Some also vitamins, minerals are all regarded as mineral, but major parts are the carbohydrates, proteins, and fats. So what is the use of carbohydrates and fats? Carbohydrates, fats provides energy, so energy to do work. So proteins are building blocks of the body. So these are the most important nutrients of our body. So now I will discuss how these acids are formed with regard to four stages. One is a normal feeding stage, second is a fasting state, exercise state, and what happens when the body undergoes some sort of disease process. So this is called a hypermetabolic or catabolic states. So let us first look at the normal feeding state. Now in normal feeding state, what when humans consumes? Carbohydrates, proteins, and fats. So these nutrients are broken down by enzymes, and during this process they release a lot of energy. So this energy is mainly ATP, and during this process, when ATP is used, so hydrogen ions are produced. So this is a person who is taking, so we take a sugar, but these are all broken down by the enzymes and all. So now we take a lunch time. So when lunch time we take a good food. This food consists of carbohydrates, proteins, and fats. So we'll now dissect what happens to our body when we take a carbohydrate, proteins after lunch and all. So for this reason, we must look at what is carbohydrates. The carbohydrates are divided into four categories: monosaccharides, disaccharides, oligosaccharides, and polysaccharides. From monosaccharides, what we find? The one sugar molecule consists of glucose, fructose, galactose. Disaccharides are two sugar molecules: sucrose, lactose, and maltose. Oligosaccharides are raffinose and stacylose, and polysaccharides are 10 or more: starch, glycose, and glucosylose. Now all these carbohydrates are broken down to simpler forms. It starts in a GIT, also cellular glands, and also intestine. After broken down, they absorb in the intestine, and they are transported to the liver. Now how is liver? Through the portal vein. This is very important. The liver, they are transported to carbohydrates, transported to help a portal vein, and what happens once it gets to the liver? The liver releases into circulation as glucose. So after taking meal, the glucose level rises because liver releases circulation, and the remaining excess is stored as glycogen. Now glucose has entered the blood from the glucose. Now this glucose, after under the effect of insulin and certain messengers, they enter the cell. Now from this glucose is then converted to pyruvate. Pyruvate is converted to the acetyl coenzyme A. Now this is really, mind you, this is in the cytoplasm of the cell. Now this acetyl coenzyme A enters the mitochondria of the cell. Now under the effect of three things, one is the oxygen, they undergo a TCA cycle or Krebs cycle to form carbon dioxide and 36 molecules of ATP. So this is called the aerobic respiration, because here oxygen is used and there is a good blood flow. So oxygen, with the help of oxygen, the carbon dioxide and 36 ATPs are formed. Mind you, once the pyruvate can be converted to the acetyl coenzyme A, but the acetyl coenzyme A cannot go back to pyruvate. So this is a very important late step of this conversion. Now once this carbon dioxide is formed, this carbon again diffuses into the blood. Now this in the blood, it reacts with water to form hydrogen ion and carbonic acid. So this is the hydrogen ion we get, which gets into the blood. So the first way how carbohydrates gives rise to acid is this way how this is formed. Now second part, how we look at the proteins. Now what are common proteins? We take eggs, meat, fish, beans, legumes, soy products. We always take in regularly on a diet. So whatever is the proteins, polymers are broken down in intestine into what? Amino acid. Now I look at the structure. Amino acid has got two parts, one is amino group and a carboxyl group. So now they transported to liver, again to the portal vein. Now in the liver it has two things. One is they create an amino acid pools. From this amino acid pools in liver, they are used to make proteins. For most important protein we make liver as a plasma protein, which has the albumin. Some hormones are also made. After making this protein, this second part of amino acid pool from the liver is released into the blood. And after releasing the blood, they are transported to the blood for manufacturing certain proteins, other proteins, tissue proteins like skeletal muscle protein, heart muscle protein. All these proteins are mainly used. Now here is the catch. Compared to glucose and fats, which we'll discuss later, which is stored in liver as glycogen and triglycerides, the excess proteins are not stored in liver. That's the most important, but have to be degraded. Once the liver uses the protein to manufacture plasma proteins, they are not stored compared to. So for this we will look at the chemistry of the amino acid. So we have looked at the chemistry, we find this amino acid had amino terminal and a carboxyl terminal and there's a carbon atom. So what happens is amino terminal, this is detoxified in liver, and when it is detoxified, it forms urea. And when this urea is converted to ammonia, and when urea and ammonia form, a lot of hydrogen ions are formed, and these are then released into the blood. So how this ammonia group ultimately forms the hydrogen ion. There are certain other amino acids like sulfur containing amino acids. They also are metabolized in liver to form sulfuric acid, and also some other amino groups like proteins like phosphoproteins, they give rise to phosphoric acid. So this is ultimately another source of hydrogen ion. What happened to the carboxyl group? They enter a tricyclic acid cycle or the Krebs cycle to form carbon dioxide. Again the carbon dioxide diffuses into the blood to form. So these are three ways how the amino acid gives rise to the acid. Now the third is the fat metabolism. Now the sources of fats we commonly take are the saturated fat. This comprises 3 to 5%, and this comes from animal sources like chicken, cheese, ice cream. So these are saturated fat. These are not so healthy fats. The second is the fourth is unsaturated fat, which is called 8 to 10%. So this includes the monosaturated fats like avocados, nuts, seeds, polyunsaturated sunflower, walnuts. These are the healthy fats. And the last fats are the trans fats. These are absolutely unhealthy fats. These fried foods, fast food, these are not so, they never should be taken in excess. So what happens to the fats in the intestine? Now these fats molecules are broken down into fatty acids and glycerol. Now they transport to the lymphatic system. They are not a portal system compared to the carbohydrates and protein. From the lymphatic system, what happens is a major part goes to systemic circulation, and then they are stored in muscle and adipose tissues. And then after that, it is transferred to liver, where it is in excess part is stored. These fatty acids are stored in the form of triglycerides. So, but what happens suppose in between meals? What happens to the fat when we are not taking food? The glucose levels are low, as in between fuels, which is called post-abdominal state, or when we are doing some fasting. So what happens is that now the glucose levels are low, the fats are broken down, the triglycerides in the liver are broken down into again glycerol and fatty acids. The glycerol jumps inside the cell to form pyruvate. Now pyruvate can go two ways. One is it can go to form glucose, or it can jump into the TCA cycle forming acetyl coenzyme A, and this after that forming ATP, hydrogen ion, again the hydrogen is formed, and oxaloacetic acid. Now oxaloacetic acid then again can jump from the mitochondria, these are in the mitochondria, remember, jump from mitochondria to form glucose in the cytosol. What about the fatty acids? Now fatty acids, they're converted to acetyl coenzyme A, but as we already talked about, acetyl coenzyme cannot go back to form pyruvate. So now the acetyl coenzyme A has to go into the triacetyl cycle to form ATP, hydrogen ion, and oxalic acid. Again the same cycle gets repeated, stabilizing the glucose levels. So in summary, in fat state, what we find? The proteins are stored as amino acids, degraded water to form sulfuric acid, phosphoric acid, and urea. These ultimately forms hydrogen ions. What about glucose? Glucose is stored as glycogen. They enter the Krebs cycle to form 36 molecule ATP, along with they form hydrogen ion. Fats are stored as triglycerides. They also can enter the triglyceride cycle to form hydrogen ion. So these are ways the hydrogen ion are formed. Now let us look at fasting. So what happens in second part? Fasting can occur if suppose a prolonged period where blood glucose goes down. Suppose you're fasting for prolonged period of time. Another way the fasting can occur because suppose the cells cannot utilize glucose, even if there is plenty, the cells cannot, because suppose the condition of diabetes, so the cells cannot, the glucose cannot enter the cells. So in all the situation, the first part is the initial fasting for 8, suppose the fasting for 8 to 10 hours. So what happens is that the glycogen in liver, which is around 70 to 80 100 grams, are now broken down. So when they're broken down to glucose 6-phosphate is converted to glucose. This process is called glycosanolysis, and then this glucose can enter the triglyceride cycle to form ATP and hydrogen ion concentration. Now when after 24-40 years, now this becomes a little bit complicated. Now after 24 to 40 years, glycogen stores gets depleted. Now what happens is that from the protein, muscles are broken down. From the proteins, muscle into amino acids. Now from this amino acids are now taken up by the liver, and they form pyruvate. Pyruvate is converted by the glucose 6-phosphate as glucose, or it can enter again the acetyl coenzyme A TCA cycle to form ATP, oxalic acid, and in oxalic acid can again be converted under the glucose 6-phosphate to glucose. Now when after 48 hours this becomes a little bit of a very cranking situation. Now the fats are broken down. Now first carbohydrate, now glycogen, now initially now the fats are broken down, triglycerides are broken down into glycerol and fatty acids. We already talked. The glucose levels are absolutely low because now we are doing a 48-hour fasting and on. So glycerol then enters a cycle to form pyruvate. Now pyruvate can go to form the glucose molecule, or it can be converted to acetyl coenzyme to form the ATP, hydrogen and oxalic acid. Oxalic acid can again go to form the glucose molecule. Now what about the fatty acid? Now this is the most important thing. Fatty acids enters to form the acetyl coenzyme. Now it can go upstream to form pyruvate. This is a rate limiting step. So now the acetyl coenzyme will have to be converted to ATP, hydrogen and oxalic acid. But the oxalic acetic acid slowly gets depleted because a lot of glucose molecules need to be formed because the patient is under fasting, so glucose has to be maintained. So once it gets depleted, the acetyl coenzyme begins to accumulate because fatty acids are now converted to acetyl coenzyme. Now this acetyl ultimately converts to ketones, and these ketones potentially form hydrogen ion. So when after 48 hours, ketones forms the hydrogen ion concentration. Now what even the third stage between exercise stage. Now when we always do a lot of exercises, every person, so this person is running, so in exercise a lot of blood flow and oxygen are required to produce ATP, to produce energy. So in mild exercise, suppose in mild exercise we do running, walking, so it doesn't cause a lot of problem because the glucose molecule is converted to pyruvate. When glucose molecule converted, 2 molecules of ATPs are formed, and this can enter the acetyl Krebs cycle to form the ATP, carbon dioxide, and the ATP which is used up for energy purposes. But what happens is that suppose we are doing a vigorous exercise, sprinting or vigorous exercise, there will be a mismatch between blood flow to the muscles and the oxygen required. There will be a mismatch. So you see this muscle, there will be a mismatch between oxygen flow and the blood flow and the oxygen required blood flow. So what will happen here? Look at this chart again. So now what happens is that the oxygen flow to the blood will be, the requirement will be very high, and therefore the blood flow oxygen will be very less. So what happens is that now the body needs ATP, so we have to convert by the glucose to pyruvate. So because from the glucose to pyruvate, some 2 molecules of ATP are formed normally, but it needs more ATP to form. So more glucose are converted to pyruvate, but now because the oxygen level is low, now pyruvate cannot be converted to acetyl coenzyme because rate limiting steps are there because oxygen is required. So this is condition for anaerobic conditions. So the pyruvate gets accumulated, this gets converted to lactate, and lactate converts to lactic acids. So this is another way why a vigorous exercise, after we get lactic acids in our body. So now the fourth stage is suppose in a disease process. What happens in a disease process? How the acids are formed? There will always be a mismatch between oxygen demand and supply, and this happens in 3 conditions. Suppose we have a low oxygen levels in the blood for hypoxemia, which is called in various lung pathology, pneumonia. So the oxygen levels are very low in this. So this one when oxygen levels are low. Secondly, when blood flow in various states are low, for the shock states, so various shock. And third condition is that when the tissues and cells cannot utilize oxygen, as in sepsis, myocardial dysfunction. In all the situation, the oxygen level is low. So what happens is that now the glucose needing the ATP, so glucose has once convert to pyruvate, it cannot enter the acetyl CoA because oxygen levels are low. This is a chart that we cannot convert, as we already talked. So now the pyruvate gets accumulated because more glucose are formed, converted to pyruvate to form ATP. Pyruvate then is converted to lactic acid. Now lactic acid being an acid, we already talked acids are substance which donates hydrogen, so it becomes a lactate and hydrogen ion process. So this is another way where in this process this helps more lot of hydrogen ion, we get a lot of lactates in sepsis and all. So in short, in summary, this is the blood. What are the sources of hydrogen ion or acids in the blood? From the carbohydrates, we get from carbon dioxide, it forms hydrogen concentration. Protein, there is a sulfuric acid, there is a phosphoric acid which also produces hydrogen ions. Ammonia gives us to urea, and then it also produces hydrogen ion concentration. Fatty acids are converted to ketones, they also produce hydrogen ion concentration. And in low oxygen states, the anaerobic condition, lactic acids gets converted to lactate and hydrogen ion. So there's another way to say that the sources of acids in bodies are divided into two categories. One is volatile acids and non-volatile acids. Carbon dioxide is a volatile because it is removed by the lungs, whereas the non-volatile is a phosphoric acid, sulfuric acid, lactic acid, ketone, and they are not removed by the lungs. So it needs a very complex mechanism to remove it, and to make it, because it needs a very complex mechanism, so it needs a complex mechanism to stabilize the hydrogen ion concentration. So these are the most important ways how sources of acids are formed in the body. In the next lecture, I will talk about the regulation of the acids in our body, and followed by talking about metabolic acidosis. Till then, thank you so much.